Hello everyone, welcome to PW English YouTube channel. I'm Shalini Somashekar, your botany teacher. I welcome you all to the UMEED NEET English batch. In today's lecture, we are going to be discussing the chapter Plant Kingdom. As you already know, in this batch, we're going to discuss every chapter in absolute detail in just one shot. That's exactly what we'll be doing in today's class as well. Before we jump into discussion of the concepts, I want to give you like a quick analysis of how many questions can be expected from this chapter. So here is the analysis table. Here is the number of questions that have been asked from this chapter over the last few years in NEET. As you can see, the number of questions range between 1 and 5. Very rarely, just one question has been asked from this chapter, and also sometimes as many as 5 questions have been asked. But on an average, overall, you can say you can expect around 2 to 3 questions from this chapter. Here we're going to learn about the different plant groups, what are its characteristic features, what are some examples and all of that. You will learn about algae, bryophytes, pteridophytes, gymnosperms in absolute detail and a little bit about angiosperms. So of all the concepts in this chapter, the concept from which questions have been frequently asked is algae. Okay, so that is according to me the most important. Following that you have gymnosperms and then the other groups. Yeah, that's about the weightage and most important concepts from this chapter. Here is a snip of the NEET syllabus for 2025 from the NTA website. As you can see here, in the syllabus, you have salient features and classification of plants into groups like algae, bryophytes, pteridophytes and gymnosperms. So you will have to learn about its salient features and examples and all of that. Some students have questions usually asking about which textbook to refer. You should refer the old textbook or the new textbook. In the old textbook, you have an additional concept which is not part of your NEET 2025 syllabus. Towards the end of the chapter, the last concept, which is about alternation of generation, that has been deleted in your new NCERT, that has been deleted in the NEET syllabus as well. So if you have the new NCERT, well and good, you can refer that. If you have the old NCERT, you don't have to worry much about the last part where you learn about alternation of generation. Okay, so I hope that was helpful. Let's get started. In the last class, in the last botany lecture, we covered the chapter biological classification, right? Over there, we spoke about the different systems of classification. We spoke about Linnaeus's 2-kingdom classification, 3-kingdom, 4-kingdom, and R.H. Whittaker's 5-kingdom classification. Over there, we discussed extensively about kingdom Monera, kingdom Protista, and kingdom Fungi. I had told you then that there's going to be an exclusive chapter just for plants, and it's called Plant Kingdom. So in this chapter, we're going to focus on plant kingdom. Okay. So under kingdom Plantae, you have many different groups. We will learn about all of them. Let's start off by understanding how classification has been done under plant kingdom. More specifically in angiosperms. You know, under plants you have different groups. You have algae, bryophytes, pteridophytes, gymnosperms and angiosperms. Angiosperms are the most advanced group of plants. They are also the most dominant group of plants, which means most of the plants that you see on planet Earth right now are angiosperms. So in the past, many attempts have been made to classify angiosperms into different groups. In another chapter, biological classification, we've also learnt the importance of classification, why classification has to be done and all of that, right? So it's important to group organisms. Many different scientists have had many approaches in classifying organisms. So we have a couple of different types of classification under angiosperms. We'll learn about 3 such different types of classification in this chapter at the beginning, okay? The first type of classification is called artificial classification. Artificial classification, one good example would be the classification of angiosperms by Linnaeus. Okay, everybody knows Linnaeus, right? Linnaeus is also called the father of taxonomy. We've learned how he is the first person to have used binomial nomenclature and all of that. So it's the exact same Linnaeus. What he basically did was, while he was trying to classify angiosperms, he mostly focused on the morphological characters. He focused on the shape of the leaves, the color of the petals probably, characters that are easily visible to the naked eye and easy to distinguish when you're comparing. Okay, so he only mostly considered gross Superficial morphological characters. What are morphological characters? Morphological characters basically refers to the external features. Characters as simple as habit, whether it is a herb, shrub or tree, the color of the flower probably, the number and shape of leaves. From each node, how many leaves are arising, what is the shape of leaves, these are the things, okay. And also, he mostly focused on vegetative characters. You know what vegetative characters are, right? In a plant, you have vegetative parts and your reproductive parts. The root, stem and leaves are considered to be a vegetative parts, and parts that are involved in sexual reproduction like the flower, fruit and seed, they are considered to be your reproductive parts. He mostly considered vegetative parts for his classification. Even if he considered sexual parts, it was mostly about the structure and number of stamens in the and ratio. So these are the criteria that Linnaeus used to classify angiosperms in his artificial system. This has some advantages, it has some disadvantages. The advantage is that it's easy to classify because you're mostly considering only the morphological features. You don't have to cut the plant and look at its inside and see how the tissue arrangement is. You're not looking at the anatomy or anything. So it's easy to classify this way. But it has certain drawbacks. Some of the drawbacks are, firstly, just because two organisms' leaves, the plants of two leaves, look alike, look similar, if you're putting them into the same group, phylogenetically, evolutionarily, they may be very distantly related. Just because you're looking at a few morphological features, it is possible that you group very distantly related organisms into the same group. It is also possible that you separate out two closely related organisms into two distant groups because you're just focusing on a few morphological features. The second disadvantage is he mostly considered vegetative characters, and you know how vegetative characters can very easily vary according to our environment. It changes according to the environment. For example, if you consider us, our skin color is not constant. You step out in the sun for a little while, you become darker in complexion, right? It changes. But if you think about how many bones I have in my body, how are the organs in my body arranged with respect to each other, these are certain things that are constant. They do not change much. Likewise, even in plants, you have certain characters, mostly vegetative characters, that are under the influence of the environment. For example, if a plant gets sufficient nutrients, its leaves will grow probably more surface area, it will spread out. If it's not getting sufficient nutrients, probably it will not grow as much. So certain things are under the influence of the environment and they can change according to the environment. If you're using such features for classification, then it is not, it's not acceptable, right? And the problem was that he gave equal weightage to vegetative and sexual characters. You should ideally give more weightage to sexual characters because they don't change as much with the changing environment. However, vegetative characters can change, right? So these are the disadvantages. In order to overcome these disadvantages, many other types of classification or system of classification came about. The next type of classification we're going to study about is called natural system of classification. This mainly was based on the natural affinities between different organisms. Over here, they used several criteria to classify living organisms. Previously, in Linnaeus' classification, he mostly focused on morphological differences, morphological similarities. Over here, they did consider morphological features, but along with that, they also considered many other different diverse characters in plants, such as anatomical features, which is internal features. They cut inside the plant, looked at how different tissues are arranged, how is the xylem arranged, how is the phloem arranged, how is the cortex, is there a hypodermis. They looked at the internal structure of plants and also used that information for classification. They used ultrastructure, anatomy, internal features, pretty much the same thing. How does development of embryo happen? What kind of chemicals does this plant have? Does it have any specific type of alkaloids or anything that I should be, concerned about? So those sort of things they considered and used that information also to classify plants. Okay, so here it was, like I already mentioned, basically, the basis was Natural affinities that exist between different organisms. Now, what is natural affinity? You have learnt the term affinity probably in chemistry, like that one particular molecule has more affinity to another one compared to some compound B, something like that. Affinity is just getting together sort of thing. How much similarity is there? Like if in a class, if you are studying in a class that has like 20, 30 students, you will always know to find your friend. You will always make friends who has a similar mindset and you both operate at the same wavelength. You laugh at pretty much the same kind of jokes. You crack similar kind of jokes. So you have more affinity. Likewise, even in plants, whenever there is more similarity, we say that they have more affinity. So such groups which have similar affinities, natural affinities to be, with similar characters, such organisms, such plants were grouped together. That's why it's called the natural system. And this was proposed by Bentham and Hooker. George Bentham and Joseph Dalton, J.D. Hooker. Okay. Now, the third type of classification we are going to talk about is called the phylogenetic system of classification. This is currently the most accepted type of classification, and this was given by Engler and Prandtl. It's not mentioned in your textbook, so don't worry. This, along with considering morphology, anatomy, embryology, phytochemistry and all of that, also considered evolutionary relationships that exist between two different organisms. Now let's say we have to classify we have three different plants with us. Let's call them A, B and C. You have to group them. Obviously, some of them will have more affinities to each other than the third one, right? So if you have like a couple of plants that you have to group together, the best thing to do is look at your evolutionary history. You know, we all evolved from a common ancestor, right? If you consider me with two other organisms, you consider me with a butterfly, you consider me with probably a dog, you consider me with a gorilla. Of course, I'm related to a butterfly also. I'm related to an extent to a dog also. I'm related to a gorilla also, right? We all are animals, we have similarities. But when you compare how much similar I am with a gorilla, a dog and a butterfly, the similarity is more between me and a gorilla because we evolved from a common ancestor, right? The similarity between me and dog will be less than me and gorilla. Between me and a butterfly will be even less than similarities between me and a dog. So how will I find out where similarities is more? Because many of our ancestors are already extinct, right? If you have to find out about our phylogeny, our evolutionary history, fossils will play a very important role. By analyzing and utilizing whatever fossil evidences we have, it is possible to group different living organisms into convenient categories. So for phylogenetic classification, they will, wherever possible, wherever available, make use of fossil evidences. But sometimes it so happens that fossil evidence is not available. At such times, they use information from other sources. Over there, we have to study about three different types, three different sources from which you can obtain information in case you're not getting any fossil evidence. We have chemotaxonomy, we have cytotaxonomy, and we have numerical taxonomy. Okay, we'll go learn about it one by one, starting with numerical taxonomy. In numerical taxonomy, basically you can consider as many characters as you want at a time. Now let's say I have three different plants A, B and C. I'm going to list out all of the characters of these three different plants, okay? Starting with the roots. Roots are of taproot system or fibrous root system. Is the stem circular? Is the stem cylindrical? Is the stem quadrangular? Does the stem have any hair on it or is it smooth? Does it show alternate phyllotaxy or opposite phyllotaxy? Does it show reticulate venation or parallel venation? Every possible character that you can think of, you will list it all down. Whenever that character is present, you can assign numbers or codes to it. Whenever a character is present, you will put plus. If the character is absent, you will put minus. Or when that said character is present in plant A, you will note it down as 1. If it's absent, you will note it down as 0. So you're doing this for all the three plants. You have all the characters in one column, then you have columns A, B and C for three different plants. Whenever the character is present, you either assign some sort of code, some sort of symbol or number, and you feed this data into computer programs. Computer programs, after analyzing all of these, there will be special softwares that will tell you which two are closely related, which two are distantly related. Of course, when they're closely related, the number of common features would be more. In whichever more number of common characters are there, those will be assigned together. So in that way, by using numbers, symbols and codes, it is possible to classify living organisms. So numerical taxonomy is basically carried out by using computer programs. It is based on all of the observable characters. You can use as many characters as you want all at once. Okay. So numbers and codes are assigned to all the characters and the data is then processed. Each character is given equal importance here, and at the same time several characters, hundreds of characters can be considered. Okay, now let's talk about cytotaxonomy. Cytotaxonomy basically, uses information about chromosomes. How is the structure of chromosomes? How many chromosomes are present? How do the chromosomes behave? So basically, when you're talking about the structure of chromosomes, the behavior of chromosomes, number of chromosomes, anything about chromosomes, it's called cytotaxonomy. Ultimately, we have chemotaxonomy. Different plants will have different chemicals present within it. It will produce different kinds of chemicals. Some plants produce terpenes, some plants produce different kinds of alkaloids. So, for example, if you take humans, for instance, if I have a gene for, let's say, blue eye color, it's possible that in my family, within my family, there will be somebody, one of my cousins or somebody who has the same character, right? Instead of looking at somebody next door, within my same family, somebody will have that character, right? So that gene responsible for blue eye color will more likely be in my house, in my family, than in my neighbor. Very similarly, whenever there is a chemical that a plant produces, the gene that is responsible for the production of that particular chemical is more likely to be present within the close relatives of that plant compared to some other distantly related plant. Whenever you come across plants producing similar type of chemicals, you can say that they have probably evolved from a common ancestor. Okay, so why are we doing all this? We're doing all this because we don't have fossil evidence. So if you don't have fossil evidence, how can you find out if two plants are closely related, distantly related? You don't know about its ancestor. So when you analyze all of the chemicals that are present in the plant, if two plants are producing similar type of chemicals, they belong to two different species, it's likely that they have evolved from a common ancestor. So you can say that they're closely related and use that information to classify that particular group of plants. So chemotaxonomy also can be used. In chemotaxonomy, basically we study and use the information of what all are the chemical constituents that a plant possesses. Okay, so this is about the different kinds of classification systems, and if at all in phylogenetic system of classification you lack fossil evidence, what are the other sources that you can use to resolve any conflict that you have in classification? Alright, now let's talk about the different groups of plants. This is a faulty heading, let me correct it right now. This is not classification of angiosperms, this is classification of plants. Okay, so under plant kingdom, you have many different groups. We have algae and bryophytes and pteridophytes and gymnosperms and angiosperms. This is in order of advancement. If you consider all of these, algae is the most primitive, mostly found in water. Bryophytes show some advancement compared to algae. Pteridophytes are a little more advanced than bryophytes. Gymnosperms even better than pteridophytes, even more evolved. Angiosperms are the most advanced or the most evolved group of plants. So this is in order of how they have evolved. Algae are the most primitive. Angiosperms are the most advanced. Similar to how you learn in vertebrates, you have fishes. From fishes, amphibians evolved. From amphibians, reptiles evolved. Very similarly, even in plants, when you consider these groups, from algae, different groups of plants have evolved. This is the most primitive and this is the most advanced. We are going to learn about all of these groups in detail one by one, and we are going to start with algae. Okay, so yeah. What do we already know about algae? What do we already know about plants? We know that plants, when we grouped plants in the 5 kingdom classification of RH Whittaker, we already know that they are generally multicellular, they are eukaryotic, and they are autotrophic or photosynthetic organisms. This is something that we already know, right? So how are these algae? Algae are photosynthetic. We know about that already. If it's photosynthetic, capable of photosynthesis, then obviously it should bear chlorophyll, right? So they bear chlorophyll. Since it is a very primitive form of plants, it does not show differentiation into root, stem and leaves. Generally, when we talk about plants, when we think of plants, the images that pop in our head are usually of angiosperms or flowering plants, where you can see clear differentiation of different plant organs. You have root, stem, leaves, all of these different plant parts. Such parts are not differentiated in algae. Algae is very primitive. So when you don't see differentiation of plant body into root, stem and leaves, we call them thalloid. The plant body is called a thallus, and such a plant body is said to be thalloid. So their structure is very, very simple. You do not have true root, stem and leaves. You have a plant body which is not differentiated into root, stem and leaves, which we call the thallus. Okay, so the plant body is very simple and we say that it is thalloid. Obviously, they are capable of synthesizing their own food, so they are autotrophic. Where do you find algae? Algae are generally found in aquatic habitats. You can find them in fresh water, you can find them in saltwater marine habitats as well. Algae can also be found on terrestrial habitats on land, but they don't grow where the area is very dry. Wherever there is moisture, wherever the land is wet, for example, on rocks, wet rocks or moist rocks, on moist wood, in such places where there is moisture, where water is available to these plants, they grow very well over there. Okay, so mostly they're aquatic, but they can grow in terrestrial habitats as well, such as moist wood, moist soil and moist stones. If they're growing on terrestrial habitats, then moisture has to be there. Okay, next. Yeah, some of these organisms are also known to form associations with other organisms. We've already learnt about lichens in the chapter biological classification. I told you how lichen is just looks like one organism, but it's actually two. Here you have an algal component called the phycobiont and you have a fungal component called the mycobiont. This algal component, being photosynthetic, will synthesize food and give it to the fungus. In return, the fungus provides shelter and helps in absorption for the alga. So they are both friends living together, helping each other out. Very similarly, you can see a very interesting association between an alga, a very simple alga, green alga, and an animal, a mammal such as the sloth bear. This sloth bear, you know, sloth bears are very slow moving organisms. Even if a predator is like coming to eat them, they can't just quickly run and escape. So in order to avoid being spotted by the predator, they will allow algae to grow on their body. The fur looks green. So since they stay on trees, they are hanging there on trees. If there are any predators, since this organism is green in color, very similar to the foliage color, very similar to the color of the leaves, it's possible that the predator will not identify this, it will not locate it, and therefore it acts like a camouflage. So this is a very interesting example where you see a plant mutually associated with an animal like a sloth bear. Right over here, what does the sloth bear get? It is getting protection from the predators because algae is helping in camouflage. What is the algae getting in return? It's getting place to live, right? It's providing shelter for the alga. So this is an example where algae are associated with certain animals. Now, let's talk about its size or form. Algae can exist in so many different forms. Sometimes they are colonial, in which case many cells come together and live like a colony. You can see that in the case of volvox. Within volvox, you have many chlamydomonas-like cells that have formed like a colony and they are living. Each green spot that you see here with two flagella like that, they are chlamydomonas-like cells. Okay, but not chlamydomonas themselves. That is a different species. Many such single cells come together and there is division of labor and they function like one organism. This is called colonial form. Example for that is volvox. Then we have filamentous forms. In filamentous forms, you know how hyphae is filamentous. I told you filamentous means cells will lie end to end like this, one on top of another, and form a long thread-like structure. We call that the filament. We see filamentous forms like Spirogyra and Eulothrix as well. So here you can see this is one cell, this is one cell, this is one cell. They're all lying one end to end and forming a long filament. Right? I told you nostoc is filamentous, the hyphae and fungi are filamentous in the previous class. Basically, they are lying end to end and forming a long thread-like structure. We call that the filament. Some kelps, which are basically brown algae, very, very pretty plants these are. Though they are algae, you know, they can grow to as tall as 100 meters. In fact, in many parts of the world, in the oceans, they're mostly found in saltwater habitats, marine waters. Underwater, they grow so tall and it kind of makes like a forest. You call that the underwater kelp forest. These are very, very pretty to look at. Okay, and this provides such a good, ecosystem for many other fishes and other animals to live. Yeah, so if you have the time, you should look it up on YouTube and watch underwater kelp forest. They're very pretty. Okay, yeah, that's about the different variations in size and form of algae. Mostly when we think about algae, we think about those slimy things that are growing on grass, but nobody really imagines or thinks about how big an alga could be. So this is something that will blow your mind. Now let's talk about how reproduction happens in algae. In algae, reproduction can happen by vegetative means, asexual means as well as sexual means. We learn about each of them one by one, starting with vegetative reproduction. Vegetative reproduction can happen by fragmentation. I told you how some of these forms are filamentous, right? That means many cells are lying end to end. Fragmentation means what? Fragment means what? Fragment means piece. What was once one organism, let's assume due to some reason, some animal ran through it or this cell decayed and died. This single filament that was there is now broken down into two fragments, two filaments. Each one will behave like a new individual organism. That is one way in which vegetative propagation can happen, vegetative reproduction can happen in algae. Asexual reproduction happens by the formation of spores. Okay, so generally, a type of spores called zoospores are formed. We've learnt about zoospores even in biological classification under fungi. So zoospores, whenever you hear the term zoo, you should know that they are motile. So zoospores are motile spores. They will usually have two flagella. Okay, so when this spore will germinate, zoospore will germinate, it will give rise to a new thallus. Ultimately, we have sexual reproduction. In sexual reproduction, you have formation and fusion of gametes. Now, depending upon the morphology of the gametes, whether the male and female gamete are similar in appearance and size, whether one is big and one is small, which one is big, which one is small, based on all of these things, you can categorize sexual reproduction into three different types: isogamous, anisogamous, oogamous. Iso means what? Iso means similar, aniso means not similar, dissimilar. So, in isogamous, you can see how the gametes are similar. Male and female gametes are very similar in size and structure. Now, in isogamous condition, the gametes may be flagellated, may not be flagellated. If the gametes are isogamous and flagellated, such type of isogamous sexual reproduction where the gametes are flagellated and similar in size, you can see in Eulothrix, which is a filamentous alga. I've given you the example of this. Another filamentous alga is Spirogyra. Over here it is isogamous, but it is non-motile, it does not have flagella. In anisogamous, one gamete will be big, the other one is small. You can see that in Eudorina, it is a type of green alga. In Volvox and Fucus, Volvox is a green alga, Fucus is a brown alga. In these, you see oogamous type of reproduction. It is very similar to how reproduction happens in us. In us, you know the female gamete, the egg cell is very large in comparison with the sperm. Sperm has flagella, it is motile, but egg does not have egg does not have any locomotory structures, is non-motile. Very similarly, even in certain species of algae like Volvox and Fucus, you can see oogamous type of reproduction, where specifically the female is large and non-motile and the male is smaller in comparison and motile. Such a type of sexual reproduction is called oogamous type of reproduction. Now, what is the ecological significance of algae? How are they important for our environment? How are they important in our biosphere? So, you know that algae is a type of plant. So, plants perform a very, very important function in our ecosystem. They are the producers, they are giving food to every animal and every heterotrophic organism directly or indirectly. So, of course, since they produce photosynthesis, we're getting food from them. And also, as a byproduct of photosynthesis, they're going to release a lot of oxygen into the atmosphere. As a raw material for photosynthesis, they even pick up carbon dioxide, which is a greenhouse gas, from the atmosphere, right? So they are helping in removing carbon dioxide from our environment. They are helping in adding oxygen to our environment. They are also helping in providing us with food. Okay, so since these mostly live in aquatic habitats, freshwater or marine habitats, whenever they photosynthesize, they release oxygen, and the concentration of oxygen in its immediate environment in the water body will increase. As a result, it will help in respiration for some fishes and other animals that are living inside the water. So these are the three important points that you should remember for ecological significance of algae. Firstly, removing carbon dioxide from the atmosphere. Secondly, introducing oxygen to the atmosphere and also increasing the concentration of oxygen in its immediate environment, immediate surroundings, so that other animals can make use of it. Also, since it's a producer, it acts as food for many different animals. Okay, so yeah, at least half of the total carbon dioxide fixation happens due to algae. This increases the level of dissolved oxygen in aquatic environments. They are the primary producers. You know what primary producers are? Like when you talk about ecology, you have producer, consumer, decomposer. Primary producers are those organisms which are capable of synthesizing their own food. Since they synthesize their own food, they will have a lot of energy rich compounds like glucose and starch and all of that, which can be used as food by other organisms, right? So it acts as the food in the food chain for many animals. So this is the ecological significance. Talking about its economic importance, how can you derive any economic benefits from algae? We are humans, we always think about how any organism or anything in nature can be useful to us, right? How can we make use of algae for our benefit? It can be used as food. You have three different species of algae that you should remember, which is used as food: Sargassum, Porphyra, and Laminaria. This, along with many other species of algae, are consumed as food. It's consumed as a delicacy, especially in Japan, China, Korea, and those countries. And then they have many hydrocolloids. Hydrocolloids are structures or compounds that are present right outside the cell wall. These are plants. Algae are plants, so they definitely have a cellulosic cell wall. Right outside its cellulosic cell wall, just give me a minute. This is the cell wall. Also remember how these algae are aquatic organisms. Right outside the cell wall, you can find hydrocolloids being present. Hydrocolloids have a very high water holding capacity. These are hydrocolloids. I'll tell you what is the function of these hydrocolloids. There are two functions basically. These are these are generally found in aquatic habitats like seas and oceans. So it's possible sometimes that they are, there's a huge wave and this algae or plant is, transferred to the shore. It's possible for this plant to lose a lot of water, become desiccated when it's not surrounded by water. So this kind of holds the water. Hydrocolloids hold the water and make sure that the plant does not die due to lack of water or absence of water in its immediate environment. And another thing is, if that happens and next time there's another wave, it's possible that this is picked and drawn into water. Until that brief period, these hydrocolloids will help in preventing desiccation in algae. Another thing is, these are plants, they require minerals. If you talk about higher plants like angiosperms, they generally absorb water and minerals from the soil. But these are not attached to the soil. Whatever minerals and nutrients they need, they will absorb it from the water itself. So, if absorption of minerals have to happen If the concentration gradient is very steep, then absorption will happen easily. Pickup or intake of those nutrients and mineral ions will happen easily. So what these hydrocolloids do is they try and concentrate all of these nutrients over here so that it is available easily to the plant cell. If it is present in the surrounding water, its concentration will be very less. If it is accumulating in the hydrocolloids, it increases its concentration so that the plant cell can pick it up very easily. Okay, so in brown algae specifically, a type of hydrocolloid called algin is produced. In red algae, a type of hydrocolloid called carrageenan is produced. Actually, in NCERT, they've given it as carrageen. It's actually carrageenan. Okay. And apart from that, some species of red algae, such as Gelidium and Gracilaria, they also produce another type of hydrocolloid called agar. I'm sure you've heard about agar. Agar is extensively used in the field of microbiology. Whenever you want to grow certain microbes like bacteria, fungi, there is a nutrient medium that we prepare in the labs, which is kind of jelly-like. Over there, to prepare the nutrient medium and give it like a jelly-like consistency, we make use of something called agar. Or if you're somebody who likes cooking and baking, you know that even while preparing jellies and ice creams and tarts, agar is used. So that is obtained from organisms called Gelidium and Gracilaria, both of which are red algae. Now talking about chlorella. Chlorella is the name of an alga. This chlorella is rich in proteins, so it is used as a protein supplement by many different by people generally, and apparently because it has a very high nutritional value, it is even used by space travelers as a protein supplement. So that is about the economic importance of algae. Mostly we are talking about how it can be used as food. There are three species example that you should remember. We are going to talk about the hydrocolloids, carrageenan in red algae, algin is in brown algae, agar specifically from Gelidium and Gracilaria in red algae, and also how chlorella is used as a protein supplement by space travelers because of its high protein content and nutritional value. Okay, so that's about the economic importance. Now, when you talk about the further classification of algae, under algae, we have some other groups. Mostly these groups are classified keeping in mind just one criterion, which is what are the kinds of pigments that has. You know, pigments are some things, some substances or chemicals you can say or compounds you can say that have the ability to absorb specific wavelengths of light. It absorbs specific wavelength of light and reflects specific wavelength of light. Whichever wavelength of light it's reflecting, that is the color that it imparts. Okay, so different groups of algae have different combination of pigments based on which pigment is more present in more quantity. We classify algae. If chlorophyll is present in high quantity, it will appear green. If there's more fucoxanthin, it will appear brown. If there's more phycoerythrin, it will appear red. So depending upon what pigments that particular alga has, we classify them. We classify them primarily into three groups: chlorophyceae, phaeophyceae, rhodophyceae. In common terms, chlorophyceae is nothing but your green algae. Brown algae is phaeophyceae and red algae is rhodophyceae. We learn about each of them one by one, starting with green algae. What do we have to know about green algae? We know that they're commonly called green algae. How is the plant body here? The plant body, if you think about the criterion used for classification in R.H. Whittaker's 5 kingdom classification, it is mentioned that in order to be grouped under plant kingdom, it has to be eukaryotic and multicellular, but over here in plant kingdom in NCERT they've mentioned that it can be unicellular, which is contradictory. Okay, I'm mentioning this because I want you to know this. So plant body here can be unicellular, colonial or filamentous. Here are some examples. Chlamydomonas is unicellular, Volvox is colonial, I've already told you that. Eulothrix and Spirogyra, both of these are filamentous. You have a little bit more advanced type of green algae also, such as Cara. Okay. Now, I told you that the criterion for classification primarily is what are the kind of pigments that has. Every photosynthetic organism algae to angiosperms, they will for sure have chlorophyll A. Okay, chlorophyll A is absolutely essential. This will be present at any cost. Along with this, other pigments may be present. In green algae, for sure chlorophyll A will be there. Along with that, you have chlorophyll B as well. In green algae, you know, since it's eukaryotic, it will have membrane-bound organelles like chloroplast. Chloroplast is present, but the shape of the chloroplast will vary. Sometimes it is cup-shaped, sometimes it's network-like, reticulate, sometimes it's discoid, sometimes it's plate-like, sometimes it's ribbon-like and spiral. So this chloroplast structure and shape can exist in different forms. Some examples you can see right here. Over here, whatever I'm outlining right now, this whole thing is actually a chloroplast. As you can see clearly, it is cup-shaped. If you look at it here, you see how it is spiral and ribbon-like. That's why it's called, the organism itself is called Spirogyra, right? So, chloroplast can appear in, can be present in many different shapes. Okay. And another important feature in green algae is, within the chloroplast, you will have structures called pyrenoids. Okay. So, within the chloroplast, there will be structures called pyrenoids. You see this only in green algae. So, what are these basically? Pyrenoids are storage bodies. They store starch and protein, and they are present inside the chloroplast. Okay. So, pyrenoids are storage bodies. Where are they located? They are located inside the chloroplast, not in the cytoplasm, and they contain protein along with starch. How do these green algae store food? Similar to higher plants, they also store food in the form of starch, but some algae can store food in the form of oil droplets as well. How is the cell wall here? Similar to the plant cell wall, it is made up of cellulose and pectin. So, it will have two layers in the wall. The inner layer will be made up of cellulose and the outer layer will be made up of pectose, or you can say pectin. Okay, so these are the examples. In order to help you remember all of the examples for green algae, I have a mnemonic, which is, your ultra clean Volvo car calms my spirit. Over here, your sounds like Eudorina, like your Eudorina. So your is Eudorina, ultra is for Eulothrix, clean is for chlorella, Volvo is for Volvox, car is for Cara, calms is for Chlamydomonas, my nothing, spirit is Spirogyra. Okay, just this one picture of this Volvo car, which is green in color, should help you remember everything. Your ultra clean Volvo car calms my spirit. Your Eudorina, ultra Eulothrix, calm chlorella, Volvo Volvox, car Cara, calms Chlamydomonas, spirit Spirogyra. Okay, that's about green algae. Now let's talk about how reproduction happens in green algae. Same. If it's vegetative, it can happen by fragmentation. If it forms spore, then generally it forms zoospores that are flagellated. Sexual reproduction can happen either by means of isogamy, anisogamy, or oogamy. It differs from species to species. It's not like one species today decides to undergo reproduction by anisogamy and tomorrow it wants to undergo oogamy. It doesn't work like that. It's species specific. One species oogamous for the rest of its life. Then you have another species probably isogamous. It's like that. Okay. Now, let's talk about the next group of algae, which is Phaeophyceae. Phaeophyceae is commonly called brown algae. Though you can see this in freshwater habitat, majority of these brown algae are seen in marine habitats itself, in seas and oceans where there is salt water. Talking about the primary criterion used for classification, of course, like all plants, this will have chlorophyll A. But along with chlorophyll A, it has another type of chlorophyll called chlorophyll C. Okay, so it has chlorophyll A and chlorophyll C. Along with that, it has some carotenoids as well and xanthophylls. There is one specific type of xanthophyle called fucoxanthin that will give it the characteristic brown color. Okay, so this fucoxanthin is a type of xanthophyle. Now, just because you call them brown algae doesn't necessarily mean they will always have to be like dark brown color. The color of brown algae can vary. There are various shades of brown, and also sometimes they can just appear olive green. The reason in change in shades of brown in brown algae is the amount of fucoxanthin present. greater the concentration of fucoxanthin, more brown it will appear. If the fucoxanthin content is less, it will appear less brown and more green, and it will appear olive green in color in that case. Okay, so the color responsible, characteristic color responsible for brown color in brown algae is fucoxanthin. It is a type of xanthophyll, remember that. Now, talking about how they store their reserve food. Previously, we saw in green algae, it can be stored in the form of starch and in some species as oil droplets as well. In brown algae, you have a special type of storage material, reserve food material, which can be laminarin or mannitol. This is important, you should remember this, okay? What about the cell wall? Cell wall, like all plants, is cellulosic in nature. We already know that in brown algae, outside the cell wall, there is a layer of hydrocolloids coating called algin. This is something we already know. We learnt about it in economic importance. How does reproduction happen here? Same story. Fragmentation can happen. Spore formation can happen. Zoospores can form flagellated spores, and also sexual reproduction can happen by isogamy, anisogamy or oogamy. Talking about the size and form, some of these are very simple. They have very simple bodies. They are filamentous with slight branching. Such simple, slightly branched forms, example, you can see in ectocarpus. This is what ectocarpus looks like, okay? But when you take like a section, like a small part of it and look at it under the microscope, it will show this filamentous structure. These filaments are branched, very simple sort of branching you can see in ectocarpus, okay? Now, some of these forms, remember I told you about the underwater kelp forest and I told you to watch that video. I told you they are brown algae, they can grow as tall as 100 meters. They are also brown algae and they are called kelp. So you see like a huge diversity in size and form in brown algae. Remember, these are organisms. In algae, you don't have xylem and phloem. Xylem and phloem are very important for a plant because xylem and phloem is what will transport water to different parts of the plant, from the root to the leaves, and food gets transported through the phloem. Over here, since these are mostly aquatic organisms, they do not have, algae generally does not have vascular tissues. They lack xylem and phloem. But still, even though they are so tall, they are capable of surviving because they are surrounded by water. The surrounding water will enter into the plant by simple diffusion itself. If you are talking about terrestrial plants like our angiosperms and gymnosperms, water is present only in the soil that has to be transported up against gravity to the leaves. So that will require xylem and phloem. Since they are living itself in water and there's water surrounding them at all times, different parts, the higher parts also can absorb water from the surrounding itself by diffusion. So even though they do not have vascular tissues, they are capable of growing that tall and surviving. Okay. Now, in some forms, in some brown algae, since you do not have root, stem and leaves, true root, stem and leaves. Remember I told you that algae does not have true root, stem and leaves. It's not differentiated. Plant body is not differentiated into root, stem and leaves because they don't have vascular tissues. In some brown algae, you see root-like structures, stem-like structures and leaf-like structures. Root-like structure is called a holdfast. Holdfast basically performs the function of anchorage. Generally, if you consider higher plants with roots, you know that roots have an important function to anchor the plant to the substratum or the soil. Over here, holdfast does that job. Okay, so if you look at this particular plant, you can see there is a root-like structure. This is the holdfast. There will also be a stem-like structure which we call the stipe, and there will be a leaf-like structure, expanded portion, which we call the frond. So over here you have a root-like holdfast, stem-like stipe, leaf-like frond. Since these are present in water, the leaves will have to float. There will require some amount of buoyancy to float. At the end of leaves, at one end of the leaves, in many species, you can see the presence of air bladders. Okay. So here you can see in fucus, air bladder is present. This will help in providing buoyancy for the plant to float or stay afloat in water. Okay, so these are the different plants. You will have to remember all of these examples. We've already learnt about sargassum and laminaria as species that are consumed as food, algae that are consumed as food, enjoyed as delicacies in different parts of the world. Along with that, you should remember fucus and dictyota. Fucus you've already learnt under oogamous type of reproduction. Dictyota is another brown alga that you should remember. Why it's called dictyota is because you can see dichotomous branching here. See, this is one branch, this is one. Whenever it's branching, it is branching into two, right? That's called dichotomous branching. Again, this produces a branch, there will be two. Again, here branch is getting produced, there are two. Again, here there are two. Every time there will be two branches that are forming. So that's why it's called dictyota, because it shows dichotomous branching. Okay. So how do you remember this? You will remember this with this example. Sir dictates extremely lame facts. Sir is sargassum, dictates is dictyota, extremely is ectocarpus, that simple branching, that one, ectocarpus. Lame is laminaria, facts is fucus. Sir dictates extremely lame facts. Sargassum, dictyota, ectocarpus, laminaria, fucus. Okay. Now let's move on to the last group of algae, which is red algae. You call that rhodophyceae. Commonly it is called red algae. Of course, like all plants, this will also have chlorophyll A, but along with chlorophyll A, it also has chlorophyll D. And the characteristic pigment that is responsible for the production of, for appearing red color is a pigment called phycoerythrin. R-phycoerythrin it's called. A type of phycoerythrin exists even in cyanobacteria or blue-green algae. The phycoerythrin that exists in red algae is called R-phycoerythrin. Phyco relates to algae. Erythro means red. You know, red blood cells are also called erythrocytes, right? Erythro means red. So the red color pigment that you find in algae is called phycoerythrin. More specifically, it's R-phycoerythrin. Even over here, majority of the species of red algae are marine. They are found in salt waters like oceans and seas. Very, very rarely you can find them in fresh water. Okay. Yeah. You know how in oceans, plants can only survive and grow up to a level. Very deep inside the oceans, light cannot penetrate. If light cannot penetrate, photosynthesis cannot happen, and those organisms, plants cannot survive, right? Even in the deepest part of the oceans where no other plants can grow, apparently red algae can grow, because different pigments have ability to absorb different wavelengths of light. Red pigment, the phycoerythrin pigment, has the ability to absorb wavelengths of light which other organisms or other pigments fail to absorb. Okay, so if you think about the wavelengths of visible spectrum, you know that violet light wavelength is less. If wavelength is less, VIBGYOR 400 to 700, wavelength is less, that means it has more frequency, more energy, more penetration power. If this violet light penetrates into the deeper parts of the ocean, green pigment cannot absorb that light or other pigments cannot absorb that light, but red pigment absorbs that very effectively. Therefore, even at certain depths in the ocean where you don't find other types of plants, it's possible to find and spot red algae there. They grow well even in areas that are well lit, as well as at great depths where very little light will penetrate. Whatever light penetrates usually will be in the blue, violet, that region. This has the ability to absorb those wavelengths very well, so they can function and thrive even in such depths of oceans. Okay. So what is the reserve food here? The reserve food here is a form of starch called Floridian starch. The structure of this Floridian starch is very similar to aminopectin and glycogen. Okay, this was a question that was asked in NEET once. So Floridian starch structure is very similar to structure of amylopectin and glycogen. How does reproduction happen here? Vegetative reproduction can happen by fragmentation. Asexual reproduction, the difference here between asexual reproduction in the other two groups in red algae is, over here spore formation happens, but the spores are not flagellated. They are non-motile spores. Okay, important point. And also reproduction can happen by isogamous, anisogamous or oogamous, but motile gametes it happens mostly by oogamous type, but the gametes are also non-motile. One thing that you should keep in mind about red algae is they have no motile cells at all. In their life cycle itself, there are no motile cells. Even the spores are non-motile, not flagellated. Even the gametes are non-motile. This is an important point that you should remember about red algae. What are some examples of red algae? You have Porphyra, Polysiphonia, Gracilaria, Gelidium. About Gracilaria and Gelidium, we already studied that is used to obtain agar, which is used in microbiology and food industry. We also learnt about Porphyra. I told you Sargassum laminaria porphyra is consumed as food. So you've heard about this also already. Along with this, remember Polysiphonia. So how do you remember this? You can remember this by using this mnemonic: poly, prepared grass jelly, poly, Polysiphonia, prepared porphyra, grass, gracilaria, jelly, gelidium. Okay, so that's about your rhodophyceae. Here is a comparison table of different classes of algae and their characters. Mostly we are considering about how are the gametes, what are the pigments, what is the reserve food, okay, and what is the cell wall made up of. All of these things we'll go learn about it one by one. We already know chlorophyceae is nothing but green algae, phaeophyceae is brown algae, rhodophyceae is red algae. What are the major pigments? All of them have chlorophyll A. Chlorophyceae has chlorophyll A and B. Phaeophyceae has chlorophyll A, C and fucoxanthin as the characteristic pigment. Rhodophyceae has chlorophyll A, D and phycoerythrin as the characteristic pigment. Store food, here it is starch, sometimes oil droplets. In Phaeophyceae it can be laminarin or mannitol. Remember laminaria, laminarin. Okay. And in red algae it is Floridian starch. Cell wall, all of them will have cellulose, but along with cellulose, outer layer hydrocolloids will be present. In brown algae you see algin. In red algae, you see cellulose, pectin and certain polysulfate esters such as carrageenan and agar and all of that. Okay. How many flagella are present and what is the position of flagella? Whenever you have flagellated cells in green algae, generally the flagella arise from the tip, from the apex. So you can expect the flagella to be 2 to 8 in number. All of them will have the equal length and they are apical, they will arise from the tip. In brown algae, the flagellated cells are generally pyriform, which means they are pear-shaped, okay, like that. And the flagella will arise from the side, we say it is lateral. The number of flagella is usually 2, you will have one long, one short, so it is unequal. In rhodophyceae, I just told you very clearly that there are no flagellated cells, spores or gametes, no flagellated cell in its entire life cycle. So flagellated cells are absent. Where do you find them? All of them you can find in freshwater, brackish water and seawater or salt water. You know what freshwater is, rivers, ponds and all of that, lakes, all of that. Salt water is ocean, seas where the salinity is very high. Brackish water is where the river water meets the ocean. Over there, in the brackish water, the salinity is intermediate. It's not as little as river water. It's not as high as sea water. So it is intermediate. That is called brackish water. Green algae you can find mostly in fresh water and you can find in salt water, brackish water also. But red and brown algae very rarely you find in fresh water. Mostly they are seen in salt water and brackish water itself. Okay. Yeah. Now let's move on to the next group of plants, which is Bryophyta. This group is slightly more advanced than algae, but they also do not have vascular tissues and they also do not have a well differentiated plant body. They are still thalloid. What is common between algae and bryophytes is that they both lack vascular tissues and both of them are thalloid. Okay. The advancement that bryophytes have over algae is that in bryophytes, sex organs are more advanced, they are better developed and they are multicellular and jacketed, whereas that is not the case in algae. That is one advancement you have of, just 1 minute. Yeah, that was my son. So let's move on. Yeah, the other advancement is that bryophytes have moved from water to land. Algae mostly you see in aquatic habitats, but bryophytes, you see them in terrestrial habitats, but still they need moisture to survive, they need water to survive, but that's an advancement, okay? So let's move on and learn more about bryophytes. In bryophytes, under bryophytes, you have two groups of plants. One is called liverworts, the other one is called mosses. Similar to how under algae we had three further classes, chlorophyceae, phaeophyceae and rhodophyceae. Here you're learning about two of them. One is mosses and the second one is liverworts. Where do you find such bryophytes? Generally, most people when they look at bryophytes, they won't even realize it's bryophytes. Most people assume that it's algae. These bryophytes are extremely short, very short plants. Okay, they look like this. If you take a really close look at some plant that is growing in a pot, if you go and take a look at some moist rocks or some moist tree trunks, you can see bryophytes growing there. They're extremely super tiny plants, very pretty plants, and very ecologically significant, very important role they play in our ecosystem. So these generally grow in areas on land where there is moisture. So they grow in moist terrestrial habitats that are not too sunny, little bit of shady places. They grow over there. And bryophytes are called amphibians of the plant kingdom. What most students get confused here is, generally amphibians live on land, they live on water. That is why they are called amphibians, right? But here they are not called amphibians because bryophytes are aquatic and bryophytes can live on land. The reason they are called amphibians of the plant kingdom is because similar to how amphibians move to water for sexual reproduction, if you know, in amphibians like frogs, external fertilization happens. They go to water and they release their gametes in the water, and in the water medium fertilization will take place. Very similarly, even in amphibians, for sexual reproduction to take place, water is required. The male gamete swims in the film of water and goes and reaches the female gamete. Water is absolutely essential here for sexual reproduction to take place. Very similar to amphibians, that's why they call the amphibians of plant kingdom. Now, how is the plant body here? I've already mentioned they do not have true root, stem and leaves. They do not have vascular system. So they are still thalloid here. Now, this plant body may be prostrate, which means it's growing horizontal to the substratum or growing parallel to the substratum, or they could be erect, which means it is growing upright, like most plants. If it's growing horizontally, if this is the soil surface and it's growing horizontally like this, then we call it prostrate, okay? I already mentioned that they don't have vascular tissue, xylem and phloem are absent. Because there is no xylem and phloem, they lack true root, stem and leaves, they are not differentiated, that's why they are thalloid. If they don't have roots, then how is this plant fixed to the substratum, fixed to the soil or wherever it is growing? They don't have true roots with xylem and phloem. They, however, have root-like structures which we call rhizoids. Okay? The root-like structures are called rhizoids. And they have stem-like structure which we call the main axis, and small leaf-like structures are present. The main plant body in bryophytes is gametophytic. Okay? If you remember, when we studied about the 5 kingdom classification, we spoke a little bit about plant kingdom very briefly, and I told you in every plant you can see something called alternation of generation. Let's try to understand what that alternation of generation is here in bryophytes. Okay, I'll explain to you the life cycle of bryophytes. In every plant's life, two phases exist. Like in our life, there are different phases. You have newborn phase, infant stage, then you have a toddler stage, then you have childhood, then you have adolescence, then you have adulthood, all of these stages. Similarly, even in bryophytes' life, there are different phases. One phase alternates with the other phase. Okay, there are two primary phases or stages. Each one alternates with the other. How they are different from each other is in the number of sets of chromosomes they have. We'll start off with the gametophyte. Gametophyte is haploid, always haploid. And the sporophyte is diploid. Now, in different groups of plants, which is the dominant plant body will vary. Talking about bryophytes, the dominant plant body is gametophytic or haploid, we say. What that means is, if you take a bryophyte such as this one and look at its cells, in every one of those cells you will have one set of chromosomes. Then we call it gametophytic phase or gametophytic generation. If it has two sets of chromosomes, then we call it sporophyte. In bryophytes, in this main plant body that you can see, there is only one set of chromosomes. So we say that the main plant body is haploid gametophyte. Okay. Why do we call this the gametophyte? It is called the gametophyte because it produces gametes. Okay. So the gametophyte produces gametes. It produces the male gamete and the female gamete. Okay, the male gamete is haploid and the female gamete is also haploid. Once the male gamete and female gamete fuse, you get the diploid zygote. Okay, now let's understand. The reason it's called gametophyte is because it will give rise to gametes. If you're calling this gametophyte because it forms gametes, why do you think this is called a sporophyte? This is called a sporophyte because it produces spores. Okay, so this sporophyte will give rise to spores. Now, when these gametes fuse, one haploid cell fusing with another haploid cell will give us a diploid zygote. This diploid zygote will develop, undergo cell division and differentiation and give rise to the sporophyte. The spores that are formed here gives rise to the gametophyte. If the spore has to give rise to the haploid gametophyte, then what will the ploidy of the spores be? The ploidy of the spores will be haploid. Over here, when haploid gametophyte is giving rise to haploid gametes, haploid cell giving rise to haploid cells, haploid plant body giving rise to haploid cells. From haploid you're getting haploid. What kind of cell division is it? It is mitosis, equational division. If from a haploid cell you get haploid daughter cells, if from a diploid parent cell you get diploid daughter cells. If the ploidy is maintained, if it is equal to the previous generation, we call it mitosis, right? So over here, haploid plant body giving rise to haploid cells. Over here, mitosis will take place. Once this process happens, two haploid cells fuse to give rise to the zygote. This process is called syngamy or fertilization. After fertilization, a single-celled zygote is formed. This single-cell zygote will undergo mitotic cell division to give rise to a multicellular sporophyte. So, from diploid to diploid here also, mitosis takes place. Now, the sporophyte is diploid. Spores are haploid. From a diploid sporophyte, you're getting haploid spores. There's a reduction in the number of sets of chromosomes. What kind of cell division do you call this? This right here is meiosis. Reductional cell division is happening. From haploid spores will germinate and give rise to a haploid gametophyte. Again, this is one single cell. This is multicellular. Over here, from one cell to form many cells, mitotic cell division will happen. Okay? Spores are unicellular. Gametes are single cell. Zygote is one single cell. One cell. Okay? Gametophyte is multicellular and sporophyte is also multicellular. In case of bryophytes, the gametophyte is the dominant phase. the gametophyte has independent existence. The sporophyte is not independent. It is at least partially or completely dependent on gametophyte. In the gametophytic plant body, you will have specialized sex organs for the formation of gametes. The male sex organ is called antheridium. The female sex organ is called archegonium. Within the antheridium and archegonium, gametes are formed. The male gamete is biflagellated. It's called an antherozoid. The female gamete is non-motile. It is called the egg cell. Okay? So over here, whatever I have labeled in orange, that represents the gametophytic phase. Whatever I have represented in blue or cyan color over here, that represents the diploid sporophytic phase or the sporophytic generation. Okay? So this is basically what happens. The plant that you're looking at is gametophytic. It has one set of chromosomes in every cell, so it's a haploid plant body. Now within this plant body, male and female sex organs will be present. The male sex organ is called antheridium, the female sex organ is called archegonium. In some species, both male and female sex organs are present on the same plant. In some species, you have separate male thallus with antheridium and a separate female thallus with archegonium. Within the antheridium, male gamete is produced by undergoing mitosis. Male gamete is called antherozoid, that is flagellated. Within the archegonium, female gamete or the egg cell is produced by mitosis. Now this antherozoid is released. Male gametes are released. When the male gametes are released, if there is water in the vicinity, it will swim in that thin film of water and reach the archegonium. Once it reaches the archegonium, it will fuse with the egg, and within the gametophyte itself, within the archegonium itself, the zygote will form. Within the archegonium, on the gametophyte itself, the zygote will develop into the sporophyte. Sporophyte does not have independent existence. It will develop on top of the gametophyte itself. It derives its nourishment from the gametophyte itself. Eventually, some cells in the sporophyte will undergo meiotic cell division to produce spores. These spores are released. These spores are haploid. They are formed by meiosis. Once these spores germinate, you get a new gametophytic plant body. Again, this cycle will continue. Generally, when we talk about the formation of gametes, we say that during gamete formation, meiosis occurs. But here is an exception. In plants, during gamete formation, mitosis will occur. During spore formation, meiosis will occur. Here it's meiosis, here it is mitosis. Okay? So this is an overview of the life cycle. We'll understand it in much more detail as we move on. The sex organs here are multicellular. I've already told you that. The male sex organ is called the antheridium. It will produce biflagellated antherozoids, which are the male gametes. The female sex organ is archegonium. The antheridium is club shaped. It will be like this. Okay. Within this antheridium, many biflagellated antherozoids are produced and they get released. Okay. The archegonium is flask shaped. It looks like this. It will have an outer layer of protective cells. Okay. This is called the venter of the archegonium. Venter of the archegonium. This is called the neck. This is called the mouth. Okay, so this is the mouth. Opening is the mouth. This is the neck. This is the venter. Within the venter, you will have the egg cell. If there is thin film of water, it will swim in that and come inside, go and fertilize here. Zygote will form here. Within the archegonium itself, the sporophyte also will form. Okay. So the antherozooids are released into water where they come in contact with the archegonium. Antherozooids fuse with the egg to produce the zygote, which is diploid. This diploid zygote will undergo mitosis to give rise to a multicellular body called the sporophyte. The sporophyte is attached to the photosynthetic gametophyte and derives nourishment from it. In the sporophyte, some cells will undergo meiosis to give rise to spores. These spores get liberated, and when they find a suitable substratum, they will germinate and give rise to the gametophyte. Okay. Now, what is some significance of bryophytes? Since bryophytes are also photosynthetic like algae, they act like producers. Many animals consume bryophytes as food. As you can see in this image, you can see like a deer that is consuming. This is moss, okay? Whatever is growing on top of this tree trunk, you can see here. These are bryophytes, these are mosses. So, it will provide food for many herbaceous animals, mammals, birds, okay? Secondly, there is a species of bryophyte, a species of moss called sphagnum. This sphagnum is used as fuel, and also since it has a high water holding capacity, it is used as a packing material for any shipment of live material. Okay, for example, if some live tissue you have to transport from one place to another, in order to ship living material, it is used as a packing outside because it has high water holding capacity. Okay, so this is what sphagnum looks like when it's alive. This is dried sphagnum. This is used as packing material and also as fuel. Just like how you make cow dung cakes and use it as fuel for burning and all that, they make cakes out of sphagnum as well and use it as fuel. Okay, next. You know, whenever there is a barren land where you find no plants, no animals, nothing, just some rocks, there is a sequence of events that will take place, which we call succession, ecological succession. Initially, there is no plant or anything. Even in such bare lands where no plants exist, where the soil is not very suitable for growth of higher plants, where soil as such doesn't exist, it's like just big rocks, these little plants, along with the help of lichens, will colonize the surface of those rocks. It will grow on top of rocks when the rocks are moist. This will help in biological weathering of rocks. Sometimes you feel very small and you feel like you're not capable of doing tough things. You should take these mosses as an example and as an inspiration to do better. These plants, like I told you, are super tiny. They're very small plants. Despite being so small, they break things as big as rocks and help in the process of weathering. If these plants don't colonize the rocks, and if the rocks don't break down into smaller soil particles, higher plants will never be able to colonize those areas. So despite being so small, they're so significant in our ecosystem. These are mosses. Along with that, what you see here, these patchy things, these are lichens. They will secrete some acids into the rock. These will, if there is a tiny crevice, they will penetrate their rhizoids into it. And if water fills in there, and when it becomes cold, the water becomes ice, there is expanding, and then the rock will break, weathering of rock happens eventually over a very extended period of time, and soil particles will form over several years, and that will make way for higher plants to come and occupy this place. Okay, mosses along with lichens are the first organisms to colonize rocks. Yeah. Another important thing is they will prevent soil erosion. You see them growing like to a great extent on the floor of forests. Because of their tiny rhizoids, they will hold the soil particles together. Even if there's very strong wind or a heavy rain, the soil particles are held very closely by these rhizoids and they prevent soil erosion. Then, like I already mentioned, bryophytes are classified into two groups. You're going to study about two groups of bryophytes. One is liverworts, the other one is mosses. Okay, this is primitive in comparison. When you're comparing these two, this is primitive and this is advanced. Here's what liverworts look like. All of these umbrella chhatri kind of things that you're looking at, they are the sexual reproductive structures. I'll explain about them. And this is what a moss looks like. Okay. So you should use your macro lens and take these pictures. You're so pretty. Yeah. First, let's learn about liverworts. Then we'll talk about mosses. Bryophytes in general are thalloid. They do not have vascular tissue. So you don't have true root, stem and leaves. So liverwort being a bryophyte is also thalloid. Example for a liverwort is Marchantia. Over here, the thallus is said to be dorsiventral. Remember, I told you that the thallus can grow prostrate or erect. Dorsiventral basically means it is flat, the dorsal and the ventral side. It's growing parallel to the surface of the substratum. So this is dorsiventral, and they're growing very closely pressed to the substratum. What you're looking at here are Marchantia thalli. Thallus is singular, thalli is plural. Marchantia is dioecious, which means it has a separate female thallus and a separate male thallus. This is the female thallus. This is the male thallus. How do I know which is female, which is male? Look here. There is a label here called archegoniophore and antheridiophore. What does phore means? Phore, phore means it's bearing something. Archegoniophore is a stalk that will bear many archegonia here. This antheridiophore is a stalk that bears many antheridia. So the male thallus will bear only antheridia, the female thallus will bear only archegonia. Many species, both antheridia, archegonia are present on the same thallus. In that case, we call it monoecious. In Marchantia, you have two separate thalli, so we call it dioecious. Okay, so here's what the actual Marchantia looks like. What you're looking at here is a female thallus. I want you to notice how you have umbrella-like structure similar to what you see here. Okay, yeah. How does reproduction happen here? It happens asexually by fragmentation and by the formation of gemmae. What is fragmentation in this case? Again, forming pieces. Here it's not filamentous. Let's say this is one thallus. For some reason, this part of the thallus decays and splits this thallus into two parts. Okay, this will become a new organism. This is equivalent to a new organism. That is what happens in fragmentation. In some species, especially in some liverworts, you can see the formation of structures called gemmae. Gemmae looks like gems only. They are discoid. They are multicellular buds. They are present inside of cup-like structures called gemma cups. What you are looking at here, I will highlight it for you in pink. This is a gemma cup. It is attached to the thallus, and within the gemma cup, whatever you're looking at here, these are gemmae. When they are liberated, if they find a suitable substratum, it will give rise to a new thallus. Okay, so this is asexual reproduction. This is found only in some liverworts. How does sexual reproduction happen? I've explained the life cycle overall. By making use of this diagram, I will help you understand it better. Here you have two different thalli. One is male, one is female. What you're looking at here is female. What you're looking at here is the male thallus. Within the female thallus, you have archegoniophore. At the tip of the archegoniophore, you have the archegonial head, which is this umbrella-like structure. Within this archegoniophore, you have archegonia. Here is what the archegonia looks like. I told you it's flask-shaped. Over here, it's inverted flask shape like this. So you can see the egg cell being present here. Let's look at the male gamete. Male gametophyte. You have the antheridiophore, antheridial head. Within the antheridial head, you have these club-shaped antheridia. Within the antheridia, biflagellate sperms or antherozoids are released. Now these antherozoids will swim in a film of water and go and reach the mouth of the archegonium. Once it reaches the mouth, it will go into the neck, go into the venter, fertilize the egg cell. From the egg, first zygote will form. From the zygote, an embryo forms. From that embryo, you get the sporophyte. Within the sporophyte, you will have three parts: foot, seta and capsule. Foot is the lowermost part, then you have seta, which is like a stalk, and then you have a capsule. Within the capsule, cells will undergo meiosis, give rise to spores. These haploid spores will give rise to the gametophyte again. Okay, so this is what happens. Now, that's about liverworts. Let's talk about mosses. Mosses life cycle is very similar to the life cycle of liverworts. The only difference is over here in the gametophytic phase, you have two stages. One stage is called protonema stage. The other stage is called the leafy stage. Mosses are generally erect. Okay, they are not prostrate like liverworts. They are erect. Over here, after the spore germinates, immediately it will not give rise to the leafy stage. Okay, I'll show you. This is what the protonema stage looks like. This is what the leafy stage will look like. If you remember, I told you initially that these are very pretty to look at. This is what a moss looks like. Okay, but when it germinates from the spore, immediately it will not form this structure. It will form a filamentous structure that is called the protonema. So from the spore, after the spore germinates, you first will have the protonema stage, which is a small, slightly branched filamentous structure, thread-like structure. This protonema will produce some branches. From the secondary branches of this protonema, this leafy stage will arise. So after the spore germinates, first you have the protonema stage, then you have the leafy stage. Okay. So gametophyte, both are gametophytes, both are haploid, but there are two stages. The first one is called the protonema stage and the second one is called the leafy stage. Like I already mentioned, protonema directly develops from the spore. It is a creeping green branched structure. Frequently filamentous means it's thread-like. Leafy stage, it develops from the secondary protonema as a lateral bud. Here in the leafy stage, again since this is bryophyte, you do not have true root, stem and leaves. In the leafy stage, you have an axis, a central axis over here. Around the central axis, leaf-like structures are spirally arranged. Okay. Over here also you have rhizoids. Generally in liverworts, the rhizoids are unicellular. In mosses, rhizoids are multicellular and they could be branched. Okay. Yeah. So the leafy stage develops from, I've told you that, they consist of upright, slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage is what bears the sex organs. Let's see how the reproduction, sexual reproduction or alternation of generation happens in mosses. Some mosses are monoecious, some are dioecious. Funaria is a moss that is monoecious. Here I have taken example of a dioecious moss where you have a separate male thallus and a separate female thallus. This is the male thallus, this is the female thallus. Male thallus will have antheridia that will produce the biflagellated antherozoid. Female thallus will have the archegonia that will have the egg cell. Antherozoids swim in this water and reach the archegonium. Within the archegonium, this is the archegonium, this is the female gametophyte. Within the archegonium, see, archegonium is here. Okay, within the archegonium, zygote will form. Zygote will develop into a multicellular sporophyte. This sporophyte is dependent on the gametophyte. The sporophyte has three parts. Foot, it will be embedded within the archegonium itself. It will have a long stalk called the seta, and at the tip what you're looking at here, this is the capsule. The cells within the capsule will undergo meiosis and produce haploid spores. Spores germinate to give protonema first and then the leafy stage, and this will continue. Okay? Yeah. So mosses, vegetative reproduction can happen by fragmentation in the protonema stage. Okay? Sexual reproduction, sex organs, antheridia, archegonia are produced at the tip of leafy shoots in the leafy stage. After fertilization, zygote develops into the sporophyte. Sporophyte has three parts, foot, seta and capsule. Sporophytes in mosses, they are more developed compared to liverworts. Some of these mosses have mechanisms to disperse the spores also. Okay, they have structures like elaters and all formed. That is not part of your syllabus. So just remember they have some mechanism by which they can release the spores and liberate the spores. Okay. Yeah, some examples of mosses are Funaria, Polytrichum and Sphagnum. Alright. So that's about mosses. Let's move on and learn about the next group of plants, which is pteridophytes. So pteridophytes show some advancement over bryophytes. Pteridophytes for the first time in the plant kingdom show presence of xylem and phloem, vascular bundles. Also, this is the first time the plant body is well differentiated into true roots, stem and leaves. What's still common between bryophytes and pteridophytes is their need of water for fertilization. Both of them need water for fertilization. Okay, let's learn more about pteridophytes. I'm sure you've all watched this movie called Jurassic Park where there's a lot of dinosaurs. If you've noticed in the movie Jurassic Park or any movie that has something to do with dinosaurs, if you see in the background, you will not see any plants producing flowers. Flowering plants will not exist. All you see are fern-like plants, huge fern-like trees present. So, these pteridophytes have evolved at a time when dinosaurs existed on our planet and roamed our planet. So they are ancient, but not many species of pteridophytes that existed back then exist this day. Many of them have gone extinct. Most pteridophytes that we see today are not huge tree ferns. They are small ferns, very small plants, tiny plants that you can see. Okay. So similar to how we learnt in algae, there are three groups, chlorophyceae, phaeophyceae and rhodophyceae. In bryophytes, we learnt about two groups, which is liverworts and mosses. In pteridophytes, there are two groups. It includes what we call horsetails and ferns. Horsetails look something like this. Okay, so you may have noticed these kind of plants like equisetum growing as ornamental plants in resorts and in restaurants and all of those places. And this is a fern. I'm sure all of you have seen ferns. You also see it on the New Zealand cricket team's cap and their jerseys and everywhere. So that is the fern. Yeah. So like I already mentioned, this is the first group of plants that possess xylem and phloem, vascular tissues. Where do they grow? They also grow on land and there should be some amount of moisture. If you remember, I told you for fertilization, they also require water. So moisture has to be there. Too much sun is not suitable for pteridophytes to grow. So they grow in damp, shady places only. However, there are some species of pteridophytes that can flourish well even in sandy soils. Okay. Next, what are some importance of pteridophytes? Many are used as soil binders to prevent soil erosion and some of them have medicinal properties as well. So they are used in traditional medicine like Ayurveda and Chinese traditional medicine. They are pretty as you can see. So they are grown as ornamental plants. They are used in bouquet making and all of that. The difference between pteridophytes and pteridophytes and bryophytes is that in bryophytes we saw that the main plant body is a haploid gametophyte. Over here it is a diploid sporophyte. Regardless of which is dominant in every plant's life, a sporophyte and gametophyte will exist. But over here the main plant body is a sporophyte. The main plant body is differentiated very well into true root, stem and leaves because it has well developed vascular tissues. We say they are true leaves. Now, if you have to talk about its life cycle, since the main plant body is sporophyte, we'll start with the sporophyte. The sporophyte is diploid. In the sporophyte plant body, you will have true root, stem, leaves, everything. Some of these leaves, let's say, some of these leaves will have specialized structures to produce spores. These structures are called sporangia. Within the sporangia, spores are formed. The leaves that bear the sporangia are called sporophylls. Okay. In many species like the equisetum, These sporophylls will aggregate spirally arrange and form compact cone-like structures. That is called the strobilus. Cone is not mandatory, it's not found in every pteridophyte. In some species, you can find them. Okay. Now, these spores are formed. These spores are haploid. The spores will germinate to give rise to what? It will give rise to the gametophyte. Gametophyte does not have true root stem and leaves. The gametophyte is thalloid. It will appear very similar to liverworts. The gametophyte is generally heart-shaped, very small, inconspicuous. Inconspicuous means very small. Since it is the gametophyte, it will be haploid. On top of the gametophyte, you can find the presence of antheridia, and you can also find the presence of archegonia. The antheridia, as always, produces antherozoids. These antherozoids will swim in water, go and reach the neck of the archegonium, go and fertilize the egg within the archegonium. Okay. So, within this gametophyte itself, the sporophyte will form. So, if here the archegonium is present, from here itself, the sporophyte will develop. Okay, and then it will give rise to the mature plant body. Here the gametophyte has a special name. We call it the prothallus. Previously we saw in bryophytes. How do you make this go away? Okay. Yeah. Okay. We saw previously in bryophytes, the main plant body was gametophyte and sporophyte was dependent on the gametophyte. Over here, the sporophyte itself is the main plant body. Sporophyte is independent. It does not depend on the gametophyte. But the gametophyte, despite not being dominant, also has independent existence. Over here in pteridophytes, both sporophyte and gametophyte are independent. Okay. This is important. Keep it in mind. Okay. So this is basically what happens in pteridophytes. Similar to how in bryophytes you had monoecious and dioecious species, even in pteridophytes, sometimes the gametophyte, there will be a separate male and separate female. Whenever the sporophyte produces two types of spores, you will have two different types of gametophytes. Okay? We'll talk about that in a little while. For now, let's understand this. In some pteridophytes, leaves are very small. We call it microphylls if the leaves are small. You can see that in Selaginella. In 2 rows, very small leaves will be arranged. In ferns, you can see big leaves. You call that macrophylls. In some leaves, sporangia will be present. Within the sporangia, spores will form. The leaves that bear the sporangia are called sporophylls. Sometimes the sporophylls will form compact cone-like structures called strobili. I told you about this. You can see this in Selaginella and in Equisetum. Within the sporangia, spores are produced by meiosis. Once the spores germinate, they will develop into the heart-shaped gametophyte that I told you about. These gametophytes can grow only in very cool, damp and shady places. That's why you do not see pteridophytes everywhere, because generally most pteridophyte, gametophytes or prothallus have this requirement. Within this you will have antheridium as well as archegonium. We call this the prothallus. The antheridia produces antherozoids that are flagellated, that will swim in water and go and reach the mouth of the archegonium and fertilize the egg. Within this prothallus itself, you can see how this sporophyte has arisen. So, sporophyte first will be on the prothallus, but later it will have independent existence. In pteridophytes, the sporophyte itself is the dominant phase. Okay. In majority of the pteridophytes, the sporophyte will produce only one type of spore. If it produces only one type of spore, we say that it is homosporous. Homo means similar, same type of spore. In some pteridophytes, such as Selaginella and Salvinia, this is important, you must remember these examples. Selaginella and Salvinia, they produce two types of spores. Hetero means different. One type of spore is small in size. We call that microspores. They are the male spores. The other type of spore is larger. We call them the megaspores. They are the female spores. Male spores will give rise to the male gametophyte. Female spores will give rise to the female gametophyte. I already told you that gametophytes may be dioecious. It depends on how many types of spores are produced. Okay? Okay. In whichever species are heterozygous like Selaginella and Salvinia, this is very important in the context of how plants evolved. Up until this point, we saw that plants will not produce seeds. Even in pteridophytes, seeds will not form. But if you talk about gymnosperms and angiosperms, we know that gymnosperms and angiosperms do produce seeds. They are seeded plants, right? So you can see here, these heterosporous pteridophytes are acting like a link in evolution, and through these heterosporous pteridophytes, seeded plants like gymnosperms and angiosperms have evolved. What happens in gymnosperms and angiosperms during seed formation? Pollen grain is the microspore. Within the pollen grain, male gametophyte will form. Pollen is the male gametophyte. Male gametophyte is released. Microspore is released. Male gametophyte is released. But what happens to the female gametophyte? Does the female gametophyte ever get released from the gynoecium? Ever? From the ovule? Does the female gametophyte or the embryo sac get released? It doesn't get released. It is retained on the sporophytic plant itself, and that is how seed formation happens. Over here also, like a precursor to seed habit, something happens. Over here, the female gametophytes, even in these heterosporous pteridophyte species, are not released outside. Spores are not released outside. Spores do not germinate and form a gametophyte outside the body of the sporophyte. The sporophyte forms the female spores, retains the female spores within the sporophytic plant body. Within the sporophytic plant body itself, those spores will germinate to give rise to the female gametophyte. For a considerable period of time, this gametophyte, female gametophyte, will be retained on the parent sporophytic plant body itself. Over here, the zygotes will develop into the young embryo within the female gametophyte itself, similar to how it happens in seeded plants. So this very event is considered to be a precursor, or what gave rise to the seed habit in gymnosperms and angiosperms. So evolutionarily, this is a very, very significant step. Okay. Now pteridophytes are further classified into 4 classes. You have Psilopsida, Lycopsida, Sphenopsida and Pteropsida. You have to learn examples for that. Psilopsida, Psilotum. Lycopsida, Selaginella, Lycopodium. Sphenopsida, Equisetum. Pteropsida includes all your ferns like Pteris, Adiantum, Dryopteris, all of these. Okay, so remember those examples. That's all about pteridophytes. Now we have to learn about gymnosperms. The advancement that gymnosperms have over pteridophytes is that gymnosperms produce seeds that pteridophytes don't. What is common between pteridophytes and gymnosperms is that in both of them, in fact in all 3 of these, sporophyte, is dominant. Okay, this produces seeds and this doesn't require water for fertilization. Bryophytes and pteridophytes did. Gymnosperms do not require water for fertilization. Okay. Now, let's learn about what gymnosperms means. Gymnos means naked. Sperma means seeds. Unlike how angiosperms produce seeds which are covered by a fruit wall, in gymnosperms, seeds are naked, they are not covered by a fruit wall. The reason why there is no fruit wall is because there is no flower here. These are not flower producing plants. Gymnosperms example include your cycas, your Christmas tree, your pinus tree and all of that. Have you ever seen a flower on a Christmas tree? Christmas tree doesn't produce flowers, right? So they produce seeds but they don't produce flowers because they lack flowers and the ovary. The seeds are naked, not covered by an ovary wall or the fruit wall. Okay, so yeah. So in gymnosperms, you don't see herbs, you see medium-sized trees and very tall trees. In fact, there's one of the tallest trees in the world called Sequoia dendron giganteum. You can see that in Yellowstone National Park in California, like huge trees, like very tall trees. In comparison with the human that is standing here, here's how tall the tree is. So this is one of the tallest tree species to ever exist on our planet, and this is probably living for hundreds of years. So yeah, they are gymnosperms. Let's talk about the root, stem and leaves in gymnosperms. Roots are like dicot plants. It has taproot system. In some species, you can see special roots. Okay. In Pinus, you can see mycorrhizal association. Mycorrhizal association is when fungi are symbiotically associated with the roots of a plant. Myco means fungus related, rhiza means root like. When fungi are associated with the roots of higher plants like gymnosperms and angiosperms, we call it mycorrhiza. This is a symbiotic relationship where both the organisms interacting are getting benefited. Fungi being filamentous will increase the surface area available for absorption of water and minerals. In return, it will get food from the photosynthetic plant, Pinus. Okay, that is mycorrhiza. In cycas, you can see something called coralloid roots. Coralloid roots look like this. In coralloid roots of cycas, you can see a mutualistic association between blue-green algae like nostoc and anabina and the roots of cycas plant. Over there, those cyanobacteria will help in nitrogen fixation. Remember when I told you in biological classification, nostoc has something called heterocyst. It can help in biological nitrogen fixation. So, organisms that are capable of biological nitrogen fixation will symbiotically be associated with these plant roots and help in nitrogen fixation. In return, they will get food and shelter because they themselves are capable of synthesizing food. So, in genera like cycas, there are specialized roots called coralloid roots. They are associated with nitrogen fixing cyanobacteria like nostoc and anabena. That's about the root. Now let's talk about the stem. The stem may be branched or may not be branched. It is branched in case of Christmas tree, in cedrus, in pinus. You can see a lot of branching. But if you talk about cycas, it's like a coconut tree. It is a cylindrical stem. There is no branching. So in cycas, I'm sure you've seen all of these. This is an ornamental plant. This also is an ornamental plant. So here you don't see that the stem is branched. Talking about the leaves may be simple or they may be compound. Leaves are very special in gymnosperms. You mostly find gymnosperms in the temperate regions, right, where the temperature is very cold and there's a lot of snowing and all of that. So these leaves can have adapted themselves to withstand extremes of temperatures. If it's like too cold, too windy, still they can adapt themselves. Okay, and another thing is the leaves here may be simple or compound. In cycas, you have leaves like this. These are a type of leaves called pinnately compound leaves. More about what pinnately compound is, we will learn in the chapter Morphology of Flowering Plants next. For now, just remember these are called pinnately compound leaves. These will persist for several years. Okay, it is not deciduous, it does not fall off, it will persist for many years. They are capable of withstanding extremes of temperature, humidity and wind. Over here, extremes of temperature is not like too hot, it's more like too cold. Therefore, in order to prevent tearing of leaves when there's too much snowfall because of the weight, the leaf should not tear. So they are more needle-like. If it is needle-like, the snow can just slip off of it. Yeah, and it also reduces the surface area that's exposed to the sun. It reduces transpiration. They have a very thick cuticle and they have sunken stomata to reduce water loss. Sunken stomata is the guard cells being slightly lower in level than the other epidermal cells. Okay. Yeah, these are some adaptations that you can see in gymnosperm leaves to tolerate extreme climatic conditions. Now let's talk about the life cycle of gymnosperms. In gymnosperms also, okay, gymnosperms are strictly heterosporous. Every gymnosperm is heterosporous. It produces two types of spores, male microspores and female megaspores. Sporophytes are the dominant plant body. So the sporophyte is diploid. This is dominant. Some species like 𝐶𝐍𝐿𝐾𝐈𝐿, they are dioecious. You have a separate male sporophyte, separate female sporophyte. While some species like Pinus, it is monoecious. On the same plant body, you will have both male and female cones formed. What are cones first? Even over here, the sporophyte will have special leaves that will, bear the microsporangia and megasporangia. So within the sporophyte, you will have microsporophyll. Wait, we'll start from male cone and female cone. So you will have something called male cone or male strobilus and female cone or female strobilus. Basically, within each cone, you will have many sporophylls. Within the sporophylls, you will have sporangia. Okay, similar to how I showed you cones in Selaginella and Equisetum previously. Even over here, I'm sure you've seen Pinus cones. They're very nice to look at. They make art out of it. So yeah, those are cones. So the sporophyte bears the male cone and the female cone. Within the male cone, you will have specialized leaves called microsporophylls. Within the female cone, you will have megasporophyll. The microsporophyll will produce microspores. The megasporophyll, okay, the microsporophyll will have microsporangium. That is a structure which produces spores. The megasporophyll will have megasporangium. It will bear megasporangia. Within the microsporangia, microspores are formed. Within the megasporangia, megaspores are formed. The megasporangia will have outer covering called integument. Within the integument, you will have cells that are called nucellar cells. One of these nucellar cells will get differentiated, will become bigger in size and undergo meiotic cell division and form the megaspore. 4 megaspores it will form. Of the 4 megaspores, 1 megaspore will function as the female gametophyte. Now this female gametophyte, it will form the female gametophyte. This female gametophyte will bear 3 or more archegonia. Within the archegonia, you will have egg cells. Okay. Now this megasporangia or the ovule will not be released. The megaspore will not be released. Megagametophyte or the female gametophyte will not be released. It is retained by the parent sporophytic plant body itself. This is what will eventually become the seed. Okay. So you have the microspore. The microspore will give rise to the male gametophyte, which is nothing but your pollen grain. Megaspore already gave rise to female gametophyte, but it's not released. It's retained within the parent sporophyte. It is retained within the sporophytic plant body. Now this pollen grain is getting released. Pollen grain is the male gametophyte. Within the pollen grain you have very few cells. You have the male gametes as well. Okay. Within the female gametophyte you have archegonia. The male gamete pollen grain will form a pollen tube and release the male gamete into the mouth of the archegonia. The male gamete will go and fuse with the egg cell. So the male gamete and the female gamete will fuse. Okay. After this fusion happens, the zygote is formed. Where is the zygote formed? It is formed inside the megasporangium itself. Zygote is diploid. This diploid zygote will undergo development, form the embryo. The embryo is retained within the seed. When the seed is released and the seed germinates, you get a new sporophytic plant body. Okay. Male cone is diploid, female cone is diploid, microsporophyll is diploid, megasporophyll is diploid, microsporangia is diploid, megasporangia are diploid. But within the megasporangia, meiosis will happen and megaspores will be haploid, microspores will be haploid. They will give rise to gametophytes that are each haploid, then diploid, embryo will be diploid. This is basically how the life cycle of gymnosperms work. Now the thing is, the sporophyte may be monoecious, may be dioecious. If it's monoecious, example is Pinus. Dioecious gymnosperm example is Cycas. Okay. That's about the life cycle of gymnosperms. For gymnosperms, you don't need water. Pollen grains are released and the wind will carry the pollen grains to the archegonium or to the megasporangium or ovule. Okay. So here there is no requirement of waters. Ultimately, we've come to the last group, which is angiosperms. This is the most advanced type of plants. Most advanced. They produce flowers. They produce seeds that are covered by fruit wall. There are many economic importance of these angiosperms. They give us food, fodder for animals, and many medicine you obtain from these plants, and many other things they are useful for. Some of the smallest plants, angiosperms have very small, almost microscopic plants like Wolffia, and some angiosperms are super tall like eucalyptus that can grow greater than 100 meters in height. Angiosperms can be classified into two types. You have monocotyledonous plants and you have dicots. This classification depends on how many cotyledons are present in the seed. In the seed, if you have one cotyledon, you call it monocots. If you have two cotyledons in the seed, you call it dicot. Okay. I just forgot to tell you one thing about gymnosperms. In gymnosperms, sporophyte is independent. Gametophyte has no free living. It's not free living. It has no independent existence. Okay, remember that. Yeah. And angiosperms, we are not learning much about it. There are 3 chapters dedicated just for angiosperms. We learn about its morphology in one chapter, its anatomy in one chapter, its life cycle, how sexual reproduction happens in another chapter. So in this chapter, you're not focusing much on angiosperms. That's all we have to do in plant kingdom. I hope all the concepts are clear and you've understood everything really well. I'll see you again next week with the chapter Morphology of Flowering Plants. Until then, take care of yourselves, keep learning, make yourselves, us and your parents proud, and stay happy. Thank you. Bye.